All right, so now we're going to get into the microscopic anatomy of the kidney, which we briefly kind of introduced in the last video, but we didn't, we just kind of talked about the main parts, but here we're actually going to talk about the nitty gritty, right? We're going to get into what types of cells we find, what, how that contributes to the functions of the nephron, right? Because that's really what we're focused on in this particular section is the workhorse, the nephron. So we're going to talk about the structure of the filtration membrane. We're going to talk about the structures and subdivisions in the renal corpuscles and renal tubules. We'll talk about the location of the juxtaglomerular apparatus and the cells we find there. And then we'll get into the histology of the different parts of the nephron, including the PCT, loop of Henle, DCT, and the collecting ducts. So if we think about the nephron, I've already kind of given it its title, right? I've said it's the workhorse of the kidney. It's the one doing the main job of the kidney, which is to filter the blood and form urine from the waste products. We're not going to get heavy into how this happens or the regulation of this, simply because that's a lot of physiology. But we do want to talk about the structures and how they lead to the functions. And so to do that, we have to talk at least what the functions are. So when we're forming urine, we can break that into 3 steps: filtration, reabsorption, and secretion. We can also control blood pressure here. So we're going to do that through how much water we reabsorb, how much of the salts that we keep. We also are going to be involved in the signaling for making red blood cells. And then we will also help absorb calcium in response to like different hormones like calcitonin and PTH. So the nephron, the renal corpuscle, right? Again, we're gonna talk about like this little rounded out section of the nephron, which is sometimes just called the glomerulus, the glomerular capsule. The glomerulus, again, is like a capillary knot surrounded by the capsule. Again, you can call it Bowman's or glomerular capsule. I'll probably just use, I might use either, but I'm kind of leaning in towards glomerular capsule. This is going to be simple squamous epithelium on the parietal layer of the capsule. And then on the visceral layer on the glomerulus, we're going to have what are called podocytes. And they're going to have these little pedicles. This is one of the places where we see a specific type of capillary. We're going to see a fenestrated capillary, since we need it to be a little, quote, leakier than a regular capillary. Right, and then between these pedicles, we're going to have little filtration slits, right, that are just forming gaps between them. So this is what it looks like for real, right? And you can kind of see it looks a little bit like a mess, and you might be looking at this going, okay, cool, none of this makes sense to me. That's okay, we're gonna get there. Here's one that has a capillary that's been broken. But let's go ahead and talk about the glomerulus up close. So I already mentioned that the glomerulus is going to be a modified capillary. It's going to be made of fenestrated capillary, so it has little holes. It's going to be leakier than a normal capillary. And so the whole point is that we are doing filtration. To filter, that means we have to let some stuff through. So the filtrate can pass through if the fenestrate matches with the filtration slits. We then have the filtration membrane. This is another fenestrated endothelium with a basement membrane, and then what we call foot processes of the podocytes, hence the name podocytes, right? Because think podo, like podiatrist, is a foot doctor. That's why they have those names. The membrane is going to be negatively charged, and this is going to help with its function. So we should remember this, but if not, just a brief overview, just a reminder of what a fenestrated capillary is. Right, so we have the single layer of endothelium, right? They have the fenestrations, the holes in between them, so that we can, quote, be leaky, right? And then we're surrounded by that basement membrane. Now, before we go and look at a picture, we're also going to talk about another location because these guys are like right up against, they're right up against each other. They're super involved with each other. And that is the juxtaglomerular apparatus, or the JGA for short. This is going to be what helps us regulate blood pressure. We have an area called the macula densa where we find cuboidal cells in the distal convoluted tubule, but it's going to actually fold over and touch, right, a certain spot, kind of right between where the afferent arteriole and the efferent arterioles are going to be, right? So bam, like right there. Right there, we also have juxtaglomerular cells. These are going to be smooth muscle cells that we find in the afferent arteriole. So this is what we're talking about. So here is the glomerular capsule with the glomerulus inside, right? We have our afferent arteriole, our efferent arteriole here, and we see that our proximal convoluted tubule is going off and twisting around, and we got the loop of Henle off view here, but then that distal convoluted tubule is going to wrap all the way back around, and it's going to touch right between those blood vessels, right? This is going to be that juxtaglomerular apparatus touching right between. We have the juxtaglomerular cells here, and we have the macula densa that are shown here in green. We see the podocytes that are wrapping around them. See how they have the little extensions and they almost kind of fit in together like little puzzle pieces, right? This is what's going to help with the filtrate, so that we're not just letting everything out. Very particular items are going to be allowed out, namely things that are small, like ions. So things like a sodium ion can be let out, glucose will be let out, water can be let out. The main things that do not escape from the glomerulus that stay in the bloodstream are going to be things like cells and proteins. And so if you are seeing those things in someone's urine sample, that's an indicator that something's gone wrong, because they should never filter out at this point. We get to the proximal and distal convoluted tubules. Again, they're called the convoluted because they're twisty. That's what tortuous means. Not tortuous like torture, but tortuous like they twist and turn, right? So they're convoluted because they do all these sorts of twists and turns. They're both made of simple cuboidal epithelium, but they have some differences. With the proximal convoluted tubule, we have more microvilli and a brushed border appearance, whereas in the distal convoluted tubule, less of it microvilli, less mitochondria, and it's shorter in length in comparison. If we look at the loop of Henle, we have the descending and ascending limb. The descending limb has a short, thick portion made of simple cuboidal, and then it thins out, changing from simple cuboidal to simple squamous. We then do a quick U-turn to go back upward, where we have the thin portion made of simple squamous and then the thick one made of simple cuboidal. So anything that's up closer to the convoluted tubules is going to be thicker, simple cuboidal. Anything that's dipping down towards the U shape is going to be thinner, made of simple squamous. And then the collecting duct, this is going to continue on with the distal convoluted tubule. It's not technically part of the nephron. This is where multiple nephrons will link up to basically drain the urine that's been formed from the nephron. So multiple distal convoluted tubules, meaning multiple nephrons, will empty into one single collecting duct, and then multiple collecting ducts will empty into a minor calyx, multiple minor calyx into the major calyx, and then the multiples of the major calyx into the renal pelvis, and then ureter. The collecting duct is simple squamous epithelium. We can do one last ditch effort to reclaim extra water here as well. It's going to respond to ADH, also known as antidiuretic hormone. And so this is going to allow us to basically insert aquaporin channels to pull more water out, to concentrate the urine more, right? So you can kind of do your last ditch effort right before that urine is going to basically leave the kidney and go to the bladder, in which case we can't reclaim that anymore. ADH is going to help the collecting duct do that last minute drawback of water. And so this is one of the things that will help concentrate your urine so it's more yellow, right? You're not losing too much water. You're not having to continuously rehydrate to an extreme. It's also, this is going to be where we can have an impact if we decide to drink alcohol. If we remember, alcohol has an impact by inhibiting ADH, right? Inhibiting the release of ADH. And so if you stop ADH from being released, the collecting duct does not put out all those aquaporin channels, meaning the collecting duct can't do this last minute water reclaim. And so this is why you end up having to pee a lot when you drink excessively, and why your urine seems so clear. It's not because you're so well hydrated from drinking, right? Don't lie to yourself. It's actually you're losing water because you're not able to do this last water reclamation step, right, that you would normally be able to do. And so that's one of the reasons why you can feel kind of cruddy after a night out. There's other things going on too, but one of the things that does not help is that you are dehydrated because your ADH was inhibited. And so this step here we're talking about was not happening while you were out having a night on the town. But here's those aquaporins, and this is why they're important, because they are going to help you pull water from the inside of the collecting duct, right, and pull it into the tissues to put it back in the bloodstream so we can reclaim water, right, pulling it from the urine found in the collecting duct and put it back into something like the bloodstream.